Are we recording? Okay.
So just let me display setting.
So don't do any cursing, okay?
Okay. So thank you.
Or do it in Spanish. So
In Spanish, okay, yeah, it's a diversity thing. Okay, I'm gonna minimize here the. So thank you, everybody. Thank you, Subhaya, for allowing me to share here some of the results in the lipid club. I'm gonna talk about PPAR gamma. As you can see here, this is a 3D structure. I was very proud of being able to do this gift the other day. And this is the PPAR gamma too. So for the people that they don't know me, I work in PPAR gamma. PPAR gamma, it is a lipid sensor that belongs to the family of the PPARs. These are nuclear receptor. And PPAR stands for peroxisome proliferator activity receptor. There are 3 different PPARs, alpha, beta, delta. This is delta in humans, but this is the beta, and the gamma is the third one. And from this PPAR gamma, there are 2 different isoforms, the PPAR gamma 1 and the PPAR gamma 2 that has additional 30 amino acids, like shown here in this structure in the N-terminal. But the sequence of the rest of the protein is exactly the same between the both isoforms. So these receptors, they were found identified in the 90s, and they didn't. We didn't know the natural endogenous ligands, but the characterization of the ligand binding domain, as shown here, it shows that they have hydrophobic pockets, and different type of fatty acids may bind them. When they are activated, they heterodimerize with the retinoic X receptor, and they bind specific sequences in the DNA, the PPAR response element, and they regulate a lot of genes. Most of the actions of these PPARs are described using reverse endocrinology, which is the use of weird compounds that they activate them, the exogenous ligands, and that they can force their binding to their DNA sequences and activate their transcriptional activity. For PPAR gamma, and in the field of diabetes, it is widely known 2 different ligands, that pioglitazone and rosiglitazone. They are known like TCDs, and they are approved to treat diabetes. I'm going to focus just on PPAR gamma, which is the receptor that is highly expressed in adipose tissue. But also, as you can see in these black bars here, it is expressed across the whole body in multiple tissues. In the adipose tissue, it is known that it's essential to promote the differentiation of adipocytes and to maintain adipose tissue in adult mice, as shown here with the adipose-specific knockout of PPAR gamma. And also, it is known that the activation of PPAR gamma with these exogenous ligands, it promotes a massive activation of PPAR gamma activity in adipocytes. And it is thought that TCDs mostly or only act at the level of the adipose tissue, regulating many genes involved in glucose, lipid metabolism, adipogenesis, insulin sensitivity, and the production of adipokines. Altogether, they're going to promote insulin sensitization, and this is why TCDs are good insulin sensitizers, and they are approved by the FDA to be given to patients with diabetes in order to control their glucose levels and also the lipid levels in the circulation. However, PPAR, it is known that TCD mediated activation of PPAR gamma in many other tissues. Directly, it is widely known that in the immune cells, it could regulate some activities, also in the muscle. And in clinics, it is widely known that Some of this activation of PPAR gamma drive dose-dependent side effects. And based on those side effects, their use in clinics is in somehow limited. But I'm not going to talk about those clinical effects. What I want to talk today is about disease linked to lipids. And that one is a non-alcoholic fatty liver disease, which is widely known by NAFLD. And also its progression to a more deleterious stage called non-alcoholic steatohepatitis, or NASH. This NAFLD is the main cause of chronic liver disease, has a huge prevalence worldwide, about 30%. Here in the USA, as you can see, the prevalence is estimated to be about 40%. And in patients with obesity and type 2 diabetes, that prevalence can go up to 70%. The disease progresses just with the simple accumulation of fat, as I will show you later. And this one in conditions are associated to risk factors, as I said, with this type 2 diabetes, obesity, even cardiovascular disease can go further into a stage of steatohepatitis. And that one can be driven also by certain genes, and also microbiota or environmental factors that could drive that progression. But how this thing happens is this one, everything started at the level of the hepatocytes, which are the main cell type in the liver, and which is they are gonna be handling the lipids. The lipids, they are gonna be, they are derived from the diet. They're going to be produced de novo due to the excess of glucose in the circulation and in condition of insulin resistance. And in these obese patients with insulin resistance in the adipose tissue, also we are going to have an increased flux of fatty acid from the adipocytes to the liver. In the hepatocytes, this excess flux of fatty acids may not be compensated with oxidation or export as VLDL particle. And therefore, the lipids, they are going to end up stored in within the hepatocytes, this promoting steatosis, disrupting mitochondria and ER function. And that one will drive eventually lipid stress events that it will promote the secretion of damage-associated molecular patterns of DAMs. And those are known to be good activator of the non-parenchymal cells in the liver, which are immune cells, mostly the Kaffer cells. And also they are going to activate eventually the hepatic stellate cells that they are going to drive fibrosis. The activation of the immune cell, of course, they are going to produce inflammation. And this inflammation and fibrosis is what is the feature of the progression into non-alcoholic steatohepatitis, which is more serious than just the simple steatosis that in clinic can be considered like a benign phenotype of the liver. Unfortunately, we don't have any treatment to a specific treatment approved by the FDA to treat NAFLD. Diet and exercise are good things because it will drive the reduction in body weight, improvement in insulin sensitivity, and therefore all these fluxes of fatty acid will be reduced. And that one, it has been shown to reduce steatosis and also even reduce inflammation and fibrosis. There are a couple of agonists of nuclear receptors right now in the final race to be approved by the FDA. I don't know if they are already approved or if they are gathering the last amount of data that they need to. But there are some hope here with the agonists of the thyroid hormone receptor beta, which is an activator of fatty acid oxidation. So doing that one, we can reduce the amount of fat in the hepatocytes, and also the agonist of the FXR by this derivative of bile acid, which also is known to have a positive effect. However, I said before that these TCDs, they have also good action. And one of them that is widely known is that they are anti-inflammatory agents. So they could act in activating PPAR gamma in immune cells, reducing inflammation. Also in vitro, it has been shown that they could reduce the activation of the hepatic stellate cells. So they should be antifibrotic agents. And as I said before, they are really well-known insulin sensitizer So activating PPAR gamma in adipocytes, they should reduce the flags of NIFA, and also they should reduce the insulin resistance. Therefore, they should reduce steatosis. So they should be the panacea for NAFLD. And in fact, long-term clinical trials have been used to reduce NAFLD in patients with diabetes and without diabetes. Here, I summarize the results of the long-term trials with the rosiglitazone and pioglitazone. And as you can see, there is like a nice reduction in steatosis. The reduction in fibrosis is here a subject of debate. The only thing that here in the first phase of the pioglitazone study, they look like to be a nice reduction that wasn't reproduced in the second phase. And then the reduction in steatosis doesn't look like to progress when we extend the clinical trials. So some authors, they are raising concerns that maybe there is a limitation of extension of the treatment with the TCD, as what you get in the first month of the insulin sensitization is what you get. So this super positive effect of TCDs may have some limitation. And some authors, they are raising, hey, maybe PPAR gamma is expressed in the liver, it is even increased that expression in the liver, and that one may not be good. So we questioned that one as well, and suggested that the activation of PPAR gamma by their endogenous ligands, which are fatty acids, that they are increased within the hepatocytes in NAFLD, or the activation of PPAR gamma in hepatocyte by this exogenous ligand may not be really precisely good, and they may account for the progression of NAFLD. In fact, that one was known many years ago. With the use of the hepatocyte-specific knockout of PPAR gamma, it was shown that PPAR gamma is an steatogenic factor in lipodystrophic model, in this ACIP, and also in the OB mice. As you can see, these mice that they develop steatosis in the liver by different mechanisms, when you knock out PPAR gamma in hepatocytes, that steatosis is significantly reduced. Furthermore, these studies showed that the activation of PPAR gamma by TCD in these models of steatosis exacerbated steatosis, the accumulation of fat in the liver of these mice. And the knockout of PPAR gamma in hepatocytes reduced dramatically the accumulation of fat in the liver. These mice, the ACIP, they don't have any adipose tissue, so the fat should go somewhere else. And the OB mice increase insulin sensitivity, and that one is good. Also, it was shown that the overexpression of PPAR gamma 1 and PPAR gamma 2 in the liver of normal mice, it promotes steatosis, suggesting that, yes, if this expression is increasing patients with NAFLD, maybe that one could be bad. But not, I mean, these studies, they were considered to be a mouse-specific effect, and in humans, something else should be happening. So also, this study, they were questioned because these guys, they are lipodystrophic, and the OB mice, they are hyperphagic. They eat a lot, no? So they might be ultra-physiological. So what we have tried to do in my group is to assess if the actions of PPAR gamma in hepatocytes contribute to this steatosis in normal mice, that they are fed just a simple high-fat diet. And also, we assessed it for the first time, what is the role of this PPAR gamma in hepatocytes in the progression of non-alcoholic fatty liver disease and NASH, because that one wasn't previously studied. And to do that one, we made some specific questions that we will show you now. So how we test the actions of PPAR gamma in hepatocytes? We use hepatocyte-specific PPAR gamma knockouts, but we use an inducible knockout mouse model. And to obtain this specific knockout, we inject adeno-associated viruses, the AAV8, that they are gonna go to the liver and express Cre recombinase under the action of this promoter that is specific for the hepatocytes, the tyrosine binding globulin promoter. So these vectors, they are injected in the PPAR gamma flux mice, and this one ended knocking out the expression of PPAR gamma in hepatocytes, and that one is permanent. How we induce NAFLD? So we can give them specific diets before the injection or after the injection of the knockouts and see what is the effect of the knockouts. Or because we are using an inducible, we can feed the mice before wild type mice, the PPAR gamma flux mice. They will fed before the injection with the specific diets to induce the phenotype in the liver. And then we can do some therapeutic approach, knocking out PPAR gamma in hepatocytes and combining this one with diets enriched, were supplemented with TCD. And I will show you some examples later. What type of diets we are using? So we start using the typical high-fat diet that have a lot of fat, 60% kilocalorie from fat. And if you feed this one for more than 6 weeks, you get some obesity and some level of steatosis. But this one doesn't progress much into NAFLD, as I will show you later. Then also we have tried nutrient deficient diets that this one look like to be really effective in inducing NAFLD, but these diets are so severe and they reduce obesity and even insulin resistance. I mean, those mice, they have low glucose and low insulin because they are in a very catabolic state, I guess. And the diets that we are using now, they are more complex. They have a combination of high fat from different sources. They have cholesterol in the diet, and they have also fructose that all of them, they're gonna have more toxicity. They're gonna maintain or improve or increase body weight, and they definitely develop human-like NAFLD phenotype. So using these different type of diets, we did different experiments, and the first one was to study if the knockout of PPAR gamma in hepatocytes in adult mice prevented diet-induced steatosis. So this is what we did. In adult male mice were injected with the AAVs. We generated the control mice or the knockouts, and they were fed for 14 weeks with the high-fat diets and the low-fat diets. As you can see here, this is the liver of a high-fat fed mouse. It's full of fat in the hepatocytes. The knockout of PPAR gamma prevented that one, as previously shown by in the other ACIP and the OBOB mice. And we wonder if we were regulating similar genes to those previously described that PPAR gamma should regulate in the liver. And to our surprise, we only observe a specific downregulation of CD36 and MOGAD1 in the knockout mice fed the high-fat diet. These genes are involved in fatty acid uptake. CD36 is the fatty acid translocase, and also resterification of fatty acids from monoacylglycerols to deacylglycerols. Also, these genes, they pop up in our RNA-seq that we ran after, and also identify the additional PPAR gamma specific targets that they are way more sensitive to the knockouts. Using another approach, knocking out PPAR gamma after the induction of obesity, we tested if these obese mice without PPAR gamma in hepatocytes, they are going to be responsive to the steatogenic effect of TCDs. And so what we did here is we induce obesity, then we inject the AAVs to induce the knockout, and a week later, half of the obese mice, they were treated with TCD. As you can see, TCD increased body weight. This is one of the side effects of TCDs in obese patients. And they are good insulin sensitizers. So as you can see here, they reduce insulin, glucose level, they were maintained, and insulin tolerant tests, they were improved. So that was telling us that the treatment was working. Also, what we observed is that the obese mice, that they have steatosis. When they were treated with the TCD, we could see that enhanced or exacerbation of steatosis by TCDs. And that one was completely blunted and reduced when the PPAR gamma knockout was knocked out. Here in this picture, you can see in this where the arrows is showing, this is a mouse that was likely around 60, 65 grams. And the liver was similar to that one of a low-fat mouse, as you can see here in this histology. So the effect of the TCDs, they are powerful. They are able to clear the fat from the liver when PPAR gamma is not there in the hepatocytes. And this one is shown here with the PPAR gamma target genes. So when PPAR gamma is there, the PPAR gamma target genes are increased. They are able to retain the lipids within the hepatocytes, but when we knock out PPAR gamma in the hepatocytes, TCD, they are super powerful insulin sensitizers, and they mobilize all the fat out of the liver and increase the mobilization into adipose tissue. There is and this one is has been shown in humans, there is an expansion of the subcutaneous adipose tissue, and that one is super efficient when PPAR gamma is knocked out in hepatocytes. However, these models, they are models of simple steatosis. These are early stages of NAFLD, and we were interested to study what is going on with the progression to a state of steatohepatitis, where we have severe steatosis and we start seeing fibrosis and inflammation. To study that one, we need to change the diet, and we combine this high-fat diet with cholesterol and fructose to study that one. So we did a similar approach to our first experiment. We knock out PPAR gamma in hepatocytes of adult mice, and then the mice that were fed for 24 weeks with this specific diet that it is known to induce NASH. So as shown here, you can see both, well, only male mice showed an increased expression of PPAR gamma in the liver when they were fed this specific diet. The knockout was effective. And that one, the increased expression of PPAR gamma was associated with increased weight of the liver and ALT levels, because in the knockouts, we could see that that one was reduced. Not only that one, the knockout also reduced the progression into non-alcoholic, the progression of NASH. And these are the slides from control mice for the low-fat diet or the NASH diet. In the NASH diet, you see that massive steatosis, inflammation, and even fibrosis. And in the knockout, we see a reduction of that effect. That one was quantified with the NAFLD activity score of the slides, and also with a reduction in the type of fibrosis. That one also, yep.
The body weight don't change, do they?
Yeah, it doesn't change. That's a limitation of this diet. We were sold the diet it is sold that it produces obesity, but that one happens when you use a chow diet as control diet. Chow diet, I don't like to use chow diets as control diet because they have different type of fibers, different type of amino acids, or because the protein source is different, and the type of fat is also different. So it is known that the amino acids, they can induce also the progression of the baseline in adiposity. So I think that when the people compare chow diet versus the NASH diet, you know, you have many other things going into the kitchen. So, but when you use the nutrient match low-fat diet, this specific diet doesn't increase body weight. But we will see at the end of the presentation, I will show you an example where we can induce obesity. So all these effects, they were associated with changes in the gene expression and the knockout of PPAR gamma. That was, I showed you, that was effective by qPCR. Also reduce expression of inflammatory, fibrogenic genes, and also PPAR gamma target genes. But here we ask if this knockout was going to increase the benefits of TCDs under these NASH conditions, which are different from the high-fat diets. So what we did first is to induce NASH with the HFCF, the NASH diet. And after 24 weeks, we inject the mice with the AAVs, half of the control and half of the knockout mice, they were treated with TCDs, because, but here, all the mice, they were maintained on the NASH diet, okay? So the mice treated with TCD, they are the pink, the control, or the red, they are the knockouts. You can see TCDs are a good insulin sensitizer and reduce and improve the response to insulin. And in the knockout mice, surprisingly, it even promote adiposity. So somehow TCD was working really well there. When we opened those mice, we saw that the knockout mice treated with TCD have a dramatic reduction in liver weight, and that one was quantified here. So liver weight was reduced, as well as the steatosis and the level of ALT. In the control mice, TCD, yes, could reduce ALT. This one is also one thing that you see in the patients treated with NASH patients treated with TCD. They have a reduction in ALT, but the effect on steatosis, they take a lot of time. They take longer time. The knockout of PPAR gamma in hepatocytes clearly exacerbated that effect. Here in the histology, you can see that these mice that they were maintained on the NASH diets and they were treated with TCD, we can reverse the phenotype almost to the level of the low-fat diet, and that one was associated with a reduction in some inflammatory and fibrogenic genes. However, here there is one thing, and the people in the clinic will say, well, is PPAR gamma expressed in the liver of patients? Because everybody is reading all these reviews from the clinical side saying that these effects of PPAR gamma in the liver, they are mouse dependent. And here we collaborated with our collaborator from Spain, Dr. Ramos Molina, that was able to collect hundreds of samples, liver samples from bariatric patients undergoing bariatric surgery. Okay, these patients, they were, as shown here, with the body mass index obese, and 70% of them, interestingly, they have NAFLD NASH. 30% of these patients, they have healthy liver. That one, of course, was associated with the increase in the OMIR, so it's because their diabetes is worse, so that one is what is associated with the progression of NAFLD. Interestingly, in these obese patients, the progression of NAFLD NASH was also associated with an increase in the expression of PPAR gamma and increase of PPAR gamma target gene expression. And that one is associated, or well, is in line with previous results from these that they were published now 13 years ago, where they showed that in a small population of patients with NAFLD, they have increased PPAR gamma expression as compared to the lean control mice. We are comparing to obese control humans, and I call these guys mice. No, they are humans. So our data showed that PPAR gamma is also expressed in patients with NAFLD, and maybe they are susceptible to response to TCD treatment. Okay, but what is PPAR gamma regulating in these livers? So we ran a lot of qPCRs, and we didn't see the changes that we were expecting. So we ended up going into a omics approach. We did RNA-seq in all the sample and even metabolomics. Interestingly, one of the key things that was always popping up in this table was amino acid metabolism. And it looks like that the hepatocyte PPAR gamma may negatively regulate methionine metabolism, and that one could promote NASH. In fact, the metabolism of methionine in the liver is an essential process that should be maintained to control cellular homeostasis. Methionine is going to be a donor of methyl groups because it's going to produce S-adenosyl methionine. And this one is going to provide this methyl group to all the methyl transferases that they are going to be able to methylate DNA, lipids, and even amino acids to maintain the cellular function. After giving these methyl groups, homocysteine is produced. This one can be used to produce glutathione, but the level of homocysteine, they are known to be toxic for the cell. So hepatocytes express this beta in homocysteine methyl transferase, which is going to remethylate homocysteine in methionine, okay? And that one is to prevent the toxicity. So it is known that methionine metabolism, it is downregulated in patients and in mice with NAFLD and NASH, and that one drives the increasing homocysteine. What we did is to measure the gene expression of these genes by qPCR, and we observed that all of them, they are downregulated in mice with NASH. These are the blue ones, the light blue. And the PPAR gamma knockout mice, they have an increased expression of PEMT. This one is an enzyme that is going to methylate phosphatidylethanolamine to produce phosphatidylcholine. So this is good to maintain the level of phospholipids. And also we had an increased expression of BHNT, which is beta-homocysteine methyltransferase. Okay, then in the mice where we have the reversion of NAFLD to a better stage close to the low-fat diet, we have also, in this case, that are the red one, an increase in the expression of these two enzymes, and that one was associated with a reduction of homocysteines. You know, and homocysteine, of course, was increased in the mice with NAFLD NASH. Not only that homocysteine, also we measure, we send the sample to the West Coast Metabolic Center, and they measure the different type of the PCs, the phosphatidylcholine. And phosphatidylcholine, the levels, they are reducing mice with NASH. TCD look like to reduce specific subtypes of these PCs, and the knockout of PPAR gamma that were associated with the treatment of TCD look like to revert. It seems that it reversing somehow that negative effect of TCD directly on the hepatocytes. So those things are being right now explored by my lab, how these methyltransferases could be accounting for the progression, the PPAR gamma dependent progression of NAFLD. And also another thing that I didn't show you is that these TCDs in mice with NASH, when PPAR gamma is not expressed in the hepatocytes, have like a super positive effect on adipose tissue, increasing the level of adiponectin. Yeah.
When you looked at the PC species, did you also look at the PE species correspondingly?
Yes, we got all of them from the core, and but we didn't get the accumulation of PE, like a significant accumulation. The nice downregulation was in the PCs. So the PCs.
Right. So if the methyltransferase is the responsible, don't you expect a reciprocal change in the PE species?
The PENT is the one that is gonna
Right.
Is gonna convert it. The PENT.
Right.
Because
so
the PCs are also produced by the other pathway.
So you don't think it is due to PEMT itself, the increase in the PCs?
Yes, and it was shown previously that TCD, rosiglitazone, can reduce the activity of PENT. I think that one was done in a cellular model, and it's seen that the TCD could have that specific action. Right.
So in this case, you didn't see a reciprocal changes in the PEs corresponding to the 38.4 and 38.6.
I think that we didn't get the upregulation of PEs.
Okay.
Yeah. And the last thing is that this, unfortunately, this diet didn't induce obesity and insulin resistance as we are used to obtain with the high-fat diets. Nonetheless, the positive effect of the PPAR gamma was not even associated with an improvement in body composition and insulin sensitivity, because they were even worse. So that one was telling us that PPAR gamma in hepatocytes really is pushing that phenotype of the liver. So because we didn't get the obesity side, the first thing that we tried is to induce obesity first with the regular high-fat diet, and then induce NASH. This one, if you agree with me, it will be something similar to what it happens in humans, where the simple steatosis is developed, and then we have some lifestyle modifications, and that one is pushing the progression into NAFLD. So we did this one with this experimental approach. The knockout was induced in the middle after the development of obesity. So in this case, both males and female mice with obesity, they have an increased expression of PPAR gamma in the liver. And the NASH, it didn't increase that expression, but it maintained the high expression of PPAR gamma. This one was, these are data from qPCR, and also we have the similar data from the Western blots. Also, both males and female mice with pre-established obesity develop they show increased liver weight when they were fed the NASH diet. And when we did the pathological scoring of these livers, we saw that both of them, males and females, they develop NASH. As shown here, high-fat diet by itself cannot develop NASH. It develops just steatosis. Inflammation and hepatocyte ballooning is not really developed in this feeding only a simple high-fat diet. So you need this combination of nutrients to push the mice over the line. And here you can see that the male mice, they have a reduction in the progression of NASH, as shown here with this scoring. Female, it wasn't significant. It looked like a trend, but some female, they also progress into NASH. When we look at the fibrosis, and this one, it is measured with a different staining, the Sirius red staining here. So we can see that the type of fibrosis increased in both males and female mice, but the area of the Sirius red stain area, it was increased only in control mice. And the knockouts, mice treated with this NASH diet, they don't have that induction in the fibrosis area. Nonetheless, both males and female mice, they have increased in the expression of the fibrogenic genes. And the knockouts of PPAR gamma in hepatocytes in both males and female mice reduce these genes that they are induced in the hepatic stellate cells, suggesting that PPAR gamma really is doing something. But this induction of these fibrogenic genes in female doesn't translate in a significant increase in the Sirius red stain area. So when we did the RNA-seq, we see also this gender-dependent effect in terms of PPAR gamma, sorry. In male mice, the knockout of PPAR gamma dysregulates thousands of genes and hundreds of genes in the mice fed the NASH diet. But in female mice, the knockout of PPAR gamma doesn't have much of an effect. That one is suggesting that maybe despite there is an effect in a specific set of genes, there are many other genes that they are dysregulated in males as compared to females. And when we look at the RNA-seq data and the metabolomic data, again, we observe that hepatocyte methionine metabolism may be dysregulated in these models. Specifically, we went to measure this BHNT. We observed that the level of BHNT, they were increased in the hepatocyte-specific PPAR gamma knockout, and that one was associated with a reduction in homocysteine levels, which they are induced in mice with NASH, in control mice. So this homocysteine is, as I said before, is a toxic molecule, and it could drive the upregulation or the activation of non-parenchymal cells to drive fibrosis. And this one was also in 2022 reported in Journal of Hepatology, showing that the hyperhomocysteinemia is a key feature in the progression of a non-alcoholic steatohepatitis, and that one by itself can dysregulate many functions in the hepatocytes. However, here again, we failed to maintain that obesity with the NASH diet. The NMR data show us that this NASH diet reduced adiposity and even reduced insulin resistance. So something is wrong with this diet. And what we did is change a little bit the formulation. We said maybe based also on reports from other people that say that the nature of the sources of the fat in our previous NASH diet was inducing some fibrosis in adipose tissue. We went back to our 60% kilocalories from fat, and this the type of fat is lard. We added cholesterol because we think that this one is key to induce the inflammatory phenotype of NASH, and we added fructose in the drinking water, just to have, again, the three types of nutrients. So as shown here, this diet is able to induce obesity, and this one is compared to the nutrient-matched diet. So these male mice, they go over 50 grams. The female mice also with time, they reach an obesity phenotype. Both males and females, they have glucose intolerance, increased level of insulin. And as you can see, even the knockout mice, they have worsened GTT, that which is not significant, but the level of insulin in the circulation is worse. The knockout was effective again. The PPAR gamma target genes, they were increased in both males and females. But again, we start seeing some gender-dependent effect. The levels of the liver weight was dramatically increased in male, but in female, it wasn't so much. Steatosis was increased in both of them, as well as ALT, but that effect was more dramatic in male mice. When we look at the genes, liver genes, we observe that the inflammation is significantly upregulated in male mice, but female mice, they don't have much of that upregulation, and as well as fibrosis. The fibrosis was significantly upregulated in male mice, but not in females, yes?
Jose, just asking you about the changes in body weight as opposed to liver weight. You know, one possibility is females are more efficient at storing lipid in subcutaneous fat, and that helps to protect the liver. So in that case, is body weight gain comparable, you know, relative, yeah.
Yeah, the thing is, I think it's comparable when we look at the different type of subdepots, how they increase at the end. But the key is here in this curve. You can see here the males, they go quickly into an obese phenotype in 12 weeks. The females, they need longer. Despite the increase at 12 weeks, they are just 30 grams, you know? So it looks like that likely whatever that they have the females, there is ovarian function, estrogen signaling, or whatever. So
but I want to say
using the progression in the disease.
Even when you look at that change in weight, you know, if you look at percent weight gain, you know, the females at 12 weeks go from 18 grams to over 30.
Yeah.
So they go up twofold. And the males at, you know, go from
25 to 50,
around 30, no, at the 12-week time, comparing to the control group, right? At 12 weeks, yes, they go from 30 up to around 50. So in a way, relative to baseline weight, the females are more efficient at gaining weight.
It could be, yeah.
So
we should look at that one. We have all these are the NMR data. So we
So the question is whether you're sparing the liver because, you know, they're better at storing fat, you know, and that protects the liver.
It might. Yeah. That it could be one,
nice
and it prevents the liver to go bad. And
rather than some liver specific effect of being female, it may be a whole body effect of how you store lipids.
Yes, because when in the previous study, when we induced that obesity, those mice, they were obese, and then we gave them the NASH diet, the female, they lost that progression. They progress into the NASH, you know, as maybe not at the same level that male, but they progress into NASH.
Yeah, just so
here they are totally protected.
Important to consider how the liver is reflecting whole body lipid storage effects.
Yeah.
You know, and the dimorphism may be, you know.
Yeah. It was recently
things
published,
outside the liver, huh?
Yeah, that is even it's even the day of the cycle, the estrous cycle, what it is driving the storage. So it's
understood. But I think, you know, the ability to store fat in subcutaneous tissue has some real advantages, and maybe in part, you know, including perhaps sparing the liver.
Yep.
You know.
Sure.
Yeah.
So anyway, so at the end is that we observe again that protective effect of the PPAR gamma knockout in the genes involved in fibrosis. So again, these are genes that they are not expressed by hepatocytes, they are expressed by the hepatic stellate cells, but the knockout of PPAR gamma in hepatocytes may change that communication with the non-parenchymal cells, and these non-parenchymal cells, they are not so active, and therefore they might not be developing as much fibrosis. I did this one, and then it's Marta told me I have catch like an outlier in this group, and this one is significant, but I say it doesn't matter. And this one was getting even better. So I think that this is clear that the knockout of PPAR gamma in hepatocytes has a positive effect, reducing fibrosis. And we have seen this one in every single study that we have done with this type of diets. Not only that one, it reduces also the development of other features of NAFLD, increase the positive effect of TCD treatment. So insulin sensitization, it is better if PPAR gamma action in hepatocytes are blocked. And that one, we think that it could be because PPAR gamma is a negative regulator of methionine metabolism, and this is what we are right now studying in my lab. We think that the PPAR gamma expression is not a taboo to call it, that is expressing in humans, and it shouldn't call simply controversial. I think that the people should do like a mechanistic studies in humans in order to study with the right controls what are the actions of PPAR gamma in the liver, if there is any. And also these gender-dependent effects, as Dr. Unterman was saying, it could be due to the peripheral metabolism that is improving in female, but also I would say that there might be some interactions, the direct interaction of estrogen signaling at the level of hepatocytes, and this one is worth it just to study in the future. So at the end, I just want to thank the people in my lab that has done this work in the last, I would say 5 years. Most of this has been done by Sam, who is not a postdoc in my lab anymore. He's now a senior research specialist in the cardiovascular core here at UIC, but he was extremely productive in the 3 years that he was in my lab, publishing these 3 papers, and based on these results, a senior editor in the Journal of Endocrinology invited us to submit a review of our studies and the role of PPAR gamma in the liver in NAFLD. Currently in my lab, I have a postdoc, is Dr. Sierra Cruz, that joined in the fall in 2022, and Isabella, who is going to be our graduate student that is starting now in May. And we have also 2 undergraduate students that they come to our lab. Also a special thanks to my mentor, Dr. Kinneman, and Dr. Layden for his leadership and allowing us to do this research at UIC. Our additional collaborators, Bruno Ramos Molina, who is amazing providing this data from humans, and the funding from NIH. And if you guys have any questions, thank you.
Good job. Yeah, one question I have was, how are you going to translate into humans? How would you knock out the PPAR gamma in liver specifically?
Say again, could you tell?
In patients, how are you going to specifically knock out PPAR gamma?